This is the second talk from Denmark I see in this session. So this will be a little different though, because it's not so mathematically heavy. We could get into the mathematics of it, but it's more an analogical talk about bridging fields and the potential that we can have in doing so. So this is the work that I did during my PhD at the Technical University of Denmark, or DTU, under the guidance of Professor Latson and Professor Nielsen. The title is to construct equivalent electrical circuits for various different kinds of reaction networks, be they chemical or biochemical in nature. So I will basically talk about my approach of how to do this construction and give a number of different examples that will hopefully show you the utility of this approach. So I assume, given the seminar, most of you have some exposure to reaction networks. So you can think of it as a combination of two aspects. So you have a structural aspect as to the connectivity of the network. So think of it like a graph where you have nodes and edges and which species are interacting with each other. There's also the functional aspect in terms of kinetics and dynamics as to what is the rate at which species convert from one to the other. You can represent them in form of rate loss, and there's obviously some reversibility and so on associated with it as well. So, of course, they can be extremely simple. So even a set of two reactions technically would constitute a network. However, they can also become extremely complicated, especially in the biological domain, where you have a number of different kinds of interactions. They could be direct, indirect, inhibitory, allosteric, catalytic. So all kinds of interactions that further muddy the waters and make modeling more challenging, especially from an analytical standpoint. So when I approached this, I wanted to see if we can convert this into something slightly different. I was coming from an engineering background, and I wanted to see if I could relate it to some of my engineering applications or potential uses. So the basis for doing any conversion would be that they must be equivalent. And so the natural way of doing so would be to say that the equations are identical. So let me use a laser pointer just to illustrate. So this is like the classic mechanics equation where you have an inertial term, you have a damping term, which is a portion of the velocity, and you have a spring constant term. So you have like a second order differential equation. In the electrical domain, once more, you get an extremely similar equation when you use the most simplistic elements of a resistor, an inductor, and a capacitor. So if you look at the charge on a capacitor and write the Kirchhoff's law, where how the charge changes as a function of time, you get a similar second order differential equation with the terms being the parameters of the resistance, inductance, and capacitance. So there has been a lot of work done long ago on relating these two and how you can convert from a mechanical viewpoint to an electrical viewpoint, and how that could be useful and has potential applications. It's a little harder to see how this conversion actually works in the chemical kinetic domain, because typically when you write a rate law, the function doesn't always fit into this kind of neat form. And so you need to do a little more thinking, okay, what would be my analogy when I want to convert from, let's say, a chemical reaction network into an electrical circuit. So the simplest way to think about it would be to say, okay, well, in a reaction network also we have fluxes, fluxes being the rate at which reactants are consumed or products get formed. They're, of course, affected by the stoichiometry of the reaction as to how many moles get used and so on. But you again have conservation equation, which is similar in the electrical domain, because your current can never be formed or created from nothing. And so you can see a parallel between fluxes and currents. Similarly, you can think of the charge on a capacitor as representing how much charge or potential on a capacitor is representing how much of a species exists. And so as your reactant gets depleted, you can think of the potential across a capacitor going down. And as a product gets formed, the potential of a product capacitor going up. And so this is kind of the basis of the analogy, where you have chemical species represented as capacitors, and you have the current representing the flux in terms of how the rate takes place. So this is, for example, the equivalent circuit that you devise for the simplest reaction A to B. So once again, A would be denoted or represented by this capacitor CA, B would be represented by this capacitor CB. So the way to think of a rate constant is very natural in terms of resistance, because so the higher the rate constant, the lower the resistance, because then you have an easier way to flow flux or an easier way to go for current to go from A to B. So your resistance essentially is a measure of the rate constant that governs this reaction. This is representing a current source, a current controlled current source. So this is a way to ensure that, so if you're assuming a 1:1 stoichiometry as we are here, then the current that goes through this loop is identical to the current that goes through this loop, because we must ensure that how much ever of A gets consumed, the same amount of B gets produced. So this is a way in the electrical domain to ensure that the currents in loops are proportional to the stoichiometries. The last symbol is that of voltage source. This is a little more abstract, because typically if you were considering just a single reaction, you would treat this as a 0 voltage source. However, if you have external loads or some kind of driver of a system and you're imposing like feeds or growths, then you can have this kind of external driver, which changes how the system would respond to your perturbation. So your voltage source can be changed depending on the system and the way you are running your process. But if you were just modeling a single reaction, then you would essentially set this to 0. You can do Kirchhoff's law analysis on this and show that you get the exact same results for the variation of the potential of A as a function of time, as if you were to directly solve the equivalent differential equation for the kinetic of first order A to B reaction. So this is a very modular way of extending. So for example, here I'm representing a simple parallel scheme. One I have reversible, one I have irreversible, but you can essentially see that I just need to include additional loops for more species. So I have one loop for A, one loop for R, one loop for S. I have current sources to control the ratio of currents between loops. And I have resistances to denote what the inverse of the rate constants is between these different reactions. And if you solve this using any commercially available or freely available electrical softwares, and you chart how the potential across these capacitors CA, CR, CS varies as a function of time, you get the very expected exponential kinetics, exponential decay in case of A. And you have, because of course S is irreversible, you accumulate that over time, and R goes through a peak where you form initially and then decay as you reverse back. So great. So even for simple toy models, this works wonderful. How does this actually work for an actual example? So essentially, the rest of the talk is going to be talking about various different kinds of examples, going further and further in complexity and showing that this kind of approach works well to model different systems. So if you're looking at, this is a classic example from 1930s, which was a very environmental importance at the time, where you wanted to study the ozone layer depletion and how ozone layer gets formed. So you have these 4 reactions that get mediated through a free radical oxygen intermediate, and you want to see, okay, what is the rate at which ozone gets formed? How do I change it depending on external wavelength lambda and so on? So for example, in this case, your energy source or lambda can be an external source. So if you change that, your voltage source can reflect how you impose that light stimulation. So if you solve this, the equivalent circuit, you get very well documented results, and they match with the ones that are already published in the literature. You can solve this numerically directly without using the electrical software, or you can do it using the electrical approach, and you get the same curves for the different species as a function of time. So this is great. Okay, you can work with simple models. It matches very well with expected results. But where could you actually have benefit in looking at the electrical approach? So one natural expectation we had was that it would work well for oscillatory systems or systems that have nonlinear dynamics, especially because there are a lot of tools available in the electrical domain to when things are varying, is alternating current analysis. So we wanted to see for a simple oscillating kind of system with chaotic elements, how the electrical scheme would perform. Would it be able to actually handle the system better than if you were to directly solve equations. So this is an example. There is, of course, many different examples you could choose. I think we chose this one because this had been worked on at DTU. But another classic example is that of the Belousov-Zhabotinsky reaction, where you have a change in color as you go through different cycles. The interesting part of the Olson attractor is that depending on what rate constant you choose or what conditions, environmental conditions you choose, you get very different dynamic behavior. So if you were to have a rate constant of 0.35, then you get very different behavior as compared to if you had around 0.16 or much greater than 0.35. So you wanted to see if you're able to replicate all these different dynamics through your electrical approach. And this is kind of what we do. So here I'm showing for different case or different resistances, how your pattern changes as a function of time. And you're able to see the same kinds of periodic and aperiodic patterns reflected in your analysis results. So the nice part of this was actually that it works extremely quickly. It handles it really potently. And even if you were to increase the number of reactions enormously, it works very well with oscillatory systems. So that kind of helped us say that this approach is not just of theoretical importance from a fundamental perspective, but also in terms of application, there could be benefits to using this on specific systems. Another classic autocatalytic example is that of the brucillator, where you have this example of A is to B ratio. So if here the reactants are A and B, forming the products D and D going through two intermediates. The nice thing is the initial ratio of your concentrations of A is to B determine the whole dynamic patterns that you form. So if A is to B is greater than 2:1, then you get a completely different dynamics as compared to if you were to get A is to B less than half. So this is essentially what we simulate and show that we can get all different kinds of dynamics. So here we are going from large ratios to very small ratios of A is to B, and you can see that we go from stable nodes to stable focus to center. Center happens around a ratio of half. Here, of course, we're assuming all the rate constants be 1, but you can simulate with different parameters and see how your thresholds and limits change as a function of that. So this is great. You can get it for a number of different examples. And now let's see if it actually works for even more complicated systems. And here we had to actually spend a fair amount of effort because so the example I'm going to talk about is one that's very close to that of a chemical engineer. So think of any catalytic process where you have a reactant that goes onto the surface of a catalyst, it diffuses into a catalyst, and then it's having a reaction. It could be first order or zeroth order, depending on how you model it. And then the products end up diffusing out, and so you are able to mediate this process. The complication here essentially is that because there's an exothermic or endothermic aspect of the reaction, so the temperature within the catalyst changes, which in turn affects the rate. And so if you want to precisely model how that impacts your process, then the mathematics can get quite complex. So you don't really need to look too deeply into the equation. So this is essentially the non-dimensionalized form of the general equation. And this is the isothermal case where it reduces and you can analytically solve it. But so here we actually had to use slightly more complicated elements, and the simple electrical elements don't do the job because your underlying equations are significantly more nonlinear and don't work as well as you would like. So you have to invoke higher order elements like amplifiers and so on, and you are able to solve the heterogeneous catalysis example. So this is essentially the general result where for different heats of reaction, how your rate constant changes as a function of temperatures. And this is the classical isothermal case where you, depending on what parameters you use, you get different curves. So this is actually quite, this was a new result in the sense that there had been work done classically in chemical engineering literature for zeroth order cases and how the temperature can cause runaway reactions. But to our knowledge, we had not seen in first order cases how this actually happens. And so our methodology helped us analyze and work through this process. And this was around the time when COVID was at its height. And so we were stuck at home and we were like, oh, can we try and use data from COVID pandemic and do some epidemiological studies? And would we get some interesting insight out of that? So this is a very simple model, of course. It's called the SIR model, where you have a pool of susceptible individuals. They get exposed to infected individuals. And the order is the number of individuals you need to be exposed to before you catch disease yourself. And then, of course, the infected pool turns into a recovered pool with different rate of naturally compared to the infection rate. You can write the equivalent differential equation. And here we took the data from different European countries, the first 14 days of the disease, and how the infected pool changes as a function of time. Quite interestingly, when you're looking through this, you see that they actually fall on a very defined mathematical scale, and there's a nice factor of 1/3. We actually postulated that this 1/3 has some mechanistic implications in terms of how the disease actually spreads and works. There's also some evidence supporting that in terms of cryo-EM, but it was an interesting outcome of the work that you can have mechanistic results coming from your electrical approach to a chemical or biochemical system of medical importance. Here I'm going to go into slightly more complex systems of a biological origin. Some of you might be familiar with this network because it's a very important pathway which mediates called dihydrofolate reductase, which mediates the reduction of dihydrofolate into tetrahydrofolate. It's important for purine synthesis. It has therapeutic implications because if it's downregulated, then you have conditions of anemia. And so trying to understand this pathway is of important clinical applications. So most of these systems can be modeled using first order or pseudo first order. So it's more easily handled as compared to if you were doing full nonlinear analysis. But what we wanted to see is not just dynamics of how these pools change as a function of time, but whether we can leverage the fact that there are fluxes and to understand what modes the system operates in. So you can cyclize this and form the general equivalent circuit. I should probably note, I think it happened before also, that I switched the notation from F to B, where I represent the nonlinear aspects. So that's just for the electrical audience. They know that there are different kinds of sources they can use in their representations. And so B is a nonlinear current source as compared to a linear current source. So you can cyclize this reaction network into this form of equivalent circuit. And now you want to identify what are the subpathways in this network and what is the flux through them. This is extremely easy in the electrical modality because you want to see where the current is flowing. And so you very quickly can identify which subpathways have zero current and which subpathways have positive current. And that's exactly what we did when we were able to identify 14 different pathways, and 7 of them have positive flux, 3 of them, and so many of them have zero flux. And of course, our postulate was that you would want to have the maximum flux possible. And so which of these 7 positive flux cycles had the maximum flux, and use the equivalent circuit for that and solve that to get the dynamics, which would probably be the most relevant for clinical application. The last example is that of glycolysis. Just see how I'm doing on time. Oh, not too bad. So here, this is of course a very classic pathway in terms of how you convert glucose into pyruvate, ubiquitously present in different kinds of organisms. This was quite challenging because the reaction rates are not easily expressed. And so the rate laws that you need to use can be quite complicated. You also need to account for naturally the dilution, growth, and so on. If you were thinking of like a chemostat culture of E. coli, and how do you want to model your system? So we took the simplified model published in 2014, where we are not using the entire model. But we simplify it saying certain reactions are much faster than others, and certain reactions are linear and can be considered to not have a kinetic roadblock. And so using that, you can get these simple functions, which you need to convert into an equivalent electrical circuit. Similar to how we did for the heterogeneous catalysis, you can do the same thing here, and you get an equivalent circuit. I've not reproduced it because it's extremely unwieldy to depict, but naturally, when you're doing the coding, it's an extremely simple thing of just adding one line as to how you want to code the function, and then it's able to convert it into an equivalent circuit. You can get these equivalent. The simulation can give you the fluxes as a function of time. This matches with some published results already. The interesting part here is that you stop feeding after a specified time, and you want to see how the system responds to your perturbation. So naturally, F1, F2 are the most upstream fluxes. They respond faster, whereas more downstream fluxes respond slower, and you have these interesting dynamic patterns generating. So you are able to apply your system to much more complicated setups, which can be conventionally quite challenging. So I hope I have been able to illustrate that this methodology is universally applicable. There is, of course, some challenge in converting the equivalent chemical reactions into the electrical circuit. However, once you are able to do so, then it's very easy to extend the network to include additional reactions or species just by adding single lines. And then the available tools that you already have can make solution of them quite simple. So with that, I'll just conclude and say that I've here outlined one method of converting chemical networks into electrical circuits. It's kind of useful to note that this is not an analogy that is the only way to look at it. There are other ways you can analogize between chemical systems and electrical systems. There have been some publications in that. However, I think this is probably the most intuitive, where you take the simplest capacitive element as a species and look at flux as a current. And there are very easy ways to convert from one to the other. I've shown that it's possible to find dynamics of interacting chemicals reactions of complex systems, or even the simpler systems or the more complex systems. Perhaps this biggest potential lies in dealing with oscillatory and chaotic places, because you can leverage the tools that you have available to you in the electrical domain. I was using something called Spice, which is perhaps you can all download and use if you need. There are also open source circuit analysis available, like methodologies available online. So you can play around with circuits, and for any given electrical or chemical reaction, see what the equivalent circuit would be and how it would respond to perturbation. So these are a number of different examples. Some of them are still working, but I think this is still the tip of the iceberg. There are a lot of potential other studies you could do using this methodology. And if nothing else, I hope it would be a way to bridge the electrical researchers with those of more chemical or biochemical network study, the people who are studying in the biochemical fields, and find a common platform for them to discuss and compare the results. So with that, I'd like to thank you all and acknowledge my two supervisors, Professor Lars Nielsen and Professor John Willadsen. Professor Willadsen, in fact, passed away a couple of years ago. So my papers are essentially the last papers that he published with in the field. So it was kind of an honor for me to be able to work with him. And I'd like to thank DTU and CFP for funding and for their help throughout my PhD. So thank you very much. And I look forward to your questions as well as the discussion following this talk.